Yeah, thanks, David. And this is actually in a totally different spectrum of research than what I was just mentioning. I think a lot of the emphasis in cell-free systems has really been centered around utilization of cell-free systems for protein therapeutic manufacturing. Diagnostics is an area we can come back to later in the podcast if we wish. But in terms of where the field may also have an impact, we can talk about this accelerating time to market. So one of the interesting advantages of cell-free systems is that they can indeed kind of accelerate our understanding of design space. And so we've actually been collaborating with LanzaTech in this area. I think we all know that rapid population growth, the rise in global living standards, climate change concerns have really intensified the need for sustainable, low-cost productions of biofuels and bioproducts. Many everyday products that we have come from chemicals that are made from petroleum. And, rather than petroleum, we, like many others, I think, are curious to know, can we use biology to capture carbon from the atmosphere and carbon oxides, carbon monoxide, carbon dioxide, et cetera, to make these types of chemicals? And if we could do that, could this help allow us to get to sustainable, low-cost manufacture of products? And so we've actually been working with LanzaTech to help the design process for engineering an organism that can grow on gas. These are Clostridia that can grow on industrial waste gases or biomass syngas to make carbon negative chemicals. And really, one of the key challenges is that Clostridia itself is a slow growing organism, and therefore the design build test cycles for studying biosynthetic pathways and their optimization can be on the order of months, 6 months, 9 months, 12 months, or longer, depending on the complexity of the pathway. And so we've actually found a tremendous amount of success working with LanzaTech to accelerate design and selection of biosynthetic pathways. So we've been using cell-free systems to accelerate the selection and optimization of biosynthetic pathways. One of the kind of most prominent examples now comes from a publication that came out last year in Nature Chemical Biology, where what we did is we explored multiple variations of a pathway, hundreds of pathway combinations to increase production of a specific sustainable chemical. We've actually gone on beyond that with them to now not just accelerate selection optimization of biosynthetic pathways. So basically, how do I find the best sets of enzymes to allow for a specific molecular transformation, but to actually guide strain engineering pipelines to accelerate time to market, and in particular, facilitate carbon negative manufacturing. So we have a manuscript that'll be coming out soon in this space. But again, we've seen a lot of exciting opportunities in using cell-free systems to study the biochemistry of molecular pathways for metabolic engineering.